Hi everyone, this is Dr. Priti Avari. Today we are going to see the microscopic structure of lymphatic tissue. The lymphatic system consists of groups of cells, tissues, and organs that act as a defense mechanism in our body. The major cells in this defense mechanism are lymphocytes. They react to attack by harmful substances by proliferating. Then they differentiate, mature at various lymphatic tissues to respond. B lymphocytes, T lymphocytes, and natural killer cells, and some supporting cells are present in lymphatic tissues. The T lymphocytes, they differentiate in thymus. That's why they are named after thymus. 70% of circulating lymphocytes are T lymphocytes. They have got quite long lifespan. They are responsible for cell-mediated immunity, helper T cell, cytotoxic T cell, regulatory or suppressor T cell, gamma or delta T cell. These are various types of T cells or T lymphocytes. B lymphocytes, they are the part of humoral immunity. They produce and secrete various circulating antibodies as immunoglobulins. They are named after the bursa of Fabricius in birds or bursa equivalent organs such as bone marrow and gall in humans. They account for about 30% of circulating lymphocytes. Now, before we start with actual microscopic anatomy, let us try to understand how the how the lymphocytes are traveling through the lymphatic tissues and blood circulation. So 70% of the lymphocytes, which will have both B and T lymphocytes, they are circulating through lymph or blood in the body. Then, wherever required, they enter in lymphatic tissue for immunological surveillance. After the surveillance is done, after their work is done, they enter the systemic circulation again. What happens with the remaining 30% of lymphocytes? They are destined to a specific tissue. They enter in the lamina propria of mucosa of various tracts as respiratory tract, gastrointestinal tract, and urogenital tract. There, they are the part of diffuse lymphatic system. Here the cells will be short-lived, immature, or activated cells. Now we'll see various lymphatic tissues or lymphatic organs. So we have primary lymphatic organs and secondary lymphatic organs. Now, what are primary lymphatic organs? In the early life, lymphocytes are present in the primary lymphatic organs as bone marrow, gut associated lymphatic tissue, and thymus. Lymphocytes differentiate into immunocompetent cells in these organs by a process called as antigen independent proliferation and differentiation. That means here the antigens are not present. Still, the lymphocytes get differentiated into T lymphocytes, B lymphocytes. After their maturation, these lymphocytes will enter in circulation. The secondary lymphatic organs, they include various lymph nodes, lymphatic nodules, tonsil, and spleen. T and B lymphocytes undergo antigen dependent activation in these organs to form effector lymphocytes, that is, plasma cells, lymphoblast, and memory cells. Here, the antigen is actually activating them to form effector lymphocytes and memory cells. So, antibody production will take place in these lymphatic organs. The other cells present in lymphatic tissue are called as supporting cells. They include eosinophils, basophils, monocytes, macrophages, neutrophils, dendritic cells, reticular cells, Langhans cells, epithelio-reticular cells. What is their function? They can communicate with lymphocytes. Together with lymphocytes, they play an important role in presentation of antigen to the lymphocytes and they help in regulation of immune response. So they are required in lymphoid tissue along with lymphocytes. Now actually we will begin with microscopic structure of a lymph node. Lymph nodes are small organs located along the pathway of lymphatic vessels. They are widely distributed throughout the body. They are bean shaped, encapsulated, 1 millimeter to 2 centimeter long. They filter the lymph carried by afferent lymphatics. Now, let us try to understand this picture. What is this? This is a complete lymph node which is being sectioned, and then we have done H&E staining of this complete lymph node. This is the low power view of lymph node under compound microscope. What can you make out? The lymph node is surrounded by a capsule, so it is encapsulated. The capsule is made up of connective tissue collagen fibers compactly arranged. At the periphery, you can see some lymphatic vessels are entering in the lymph node. These are called as afferent lymphatics. Within the lymph node, you can see the lymphocytes are arranged. At least you can make out there are two different regions. The outer region is cortex and the inner region is medulla. The place between them is called as paracortex. The paracortex is a thymus dependent area. That means the T lymphocytes, majority of T lymphocytes are present into this paracortex. The lymph node has got hilum where you can see the efferent lymphatics are coming out. Before we enter into details of cortex and medulla, we should understand how the lymph is circulating through the lymph node. So, first, the lymph enters in the lymph node through afferent lymphatics. So, from all various regions, lymph is carried to the lymph node by these afferent lymphatics. Then, the lymph enters into subcapsular sinus. So, very important feature of a lymph node is a space underneath the capsule called as subcapsular sinus where the lymph is entering. This is the identifying feature of a lymph node. Then you can make out the capsule is giving inside some septi. These are called as trabeculae. On both sides of trabecula, you can make out again a space. This is called as trabecular sinus. So from the subcapsular sinus, the lymph enters into the trabecular sinus. Then the trabecular sinus will enter or the trabecular sinus will continue as medullary sinus in the medulla. So you can see many spaces present in the medulla, they are connected to each other. These are all medullary sinuses. Finally, the lymph comes out of the lymph node through efferent lymphatics at the hilum. So this is how the lymph is being circulated through a lymph node. So the lymph is carrying lymphocytes. Lymph is also carrying the microbes and foreign bodies and chemical harmful substances, everything in the lymph node for filtration. At the beginning, when the lymph nodes are being formed, at that time, the first time when the lymphocytes are entering, most of them are entering in the lymph node through the blood. So I haven't drawn those blood vessels here. You need to assume that there will be some sections of blood vessels in between. Just to simplify, I just haven't included the blood capillaries, but blood capillaries are also present within the lymph node. Here, what I wanted to emphasize is that the whatever harmful substances are carried to the lymph node, basically they will be carried by the lymph, and even the lymphocytes afterwards will be carried to the lymph node by lymph. Little bit about the supporting network of tissue present in the lymph node. In a lymph node, Reticular cells and reticular fibers will form the supporting network. All cells of the lymphatic tissue are supported by this network. Now, actually, we will start with the microscopic structure of a lymph node under low power in compound microscope. So, in this diagram, you can make out a capsule is there surrounding the lymph node. Underneath that, there is subcapsular sinus, the trabeculae entering inside and dividing the cortex into, you can say, compartments. The trabecular sinus continuing as medullary sinus, and then finally the efferent lymphatics. You can make out that. Now, what is there in the cortex? The cortex is completely filled by groups of lymphocytes. Now, how can you make out lymphocytes? These blue color dots represent the nuclei of lymphocytes. We can't differentiate whether it's a B lymphocyte or T lymphocytes, but their nuclei are darkly stained, heterochromatic. So, basophilic stain is taken by the nuclei. Now, inside you can see some rounded structure. What are these rounded structures? These are called as lymphatic nodules. So, we will talk about these lymphatic nodules first, then we will move on to medulla. Lymphatic nodules are also called as lymphatic follicles. In the cortex of lymph node, lymphocytes get arranged in rounded cluster called as lymphatic nodules. There are two types of these nodules, primary and secondary. How the primary lymphatic nodule appears under microscope? You can see a lymphatic nodule made up of cluster of small lymphocytes. So you can see these blue dots. These are the nuclei of these small lymphocytes. They are very closely packed here. The nuclei are darkly stained because of dense heterochromatin in them. So this is a primary lymphatic nodule. This is not activated yet. Then there is formation of secondary lymphatic nodule. Now how it is formed? This secondary lymphatic nodule develops when a lymphocyte that has recognized an antigen returns to a primary nodule and divide to form lymphoblast and plasmablast. So in this picture you can see in the center the lymphocytes are activated, they had proliferated and they divide, they differentiate and they form plasmablast and lymphoblast. So, how it appears? It appears as a faintly stained area in the center called as germinal center. Here, the lymphoblast and plasmablast are present. These are the immature lymphocytes. Their nuclei contain dispersed euchromatin, which stains lightly basophilic. This germinal center will be surrounded by a mantle zone or corona formed by small lymphocytes, the mature lymphocytes. So when we see secondary lymphatic nodule, that means the antigen has already entered inside and it has activated the lymphocytes. What can we see in the medulla? Medulla is made up of medullary sinuses and medullary cords. Now, what are these medullary cords? The medullary cords are made up of lymphocytes, the clusters of lymphocytes arranged in the form of cords. But here the cells are loosely packed, so it appears little bit faintly stained. Let us try to elaborate the exact function of the lymph node. So with the help of diagram, we will try to understand the functions. Suppose the bacteria has entered in the circulation and they are entering in the lymph node through lymph. These bacteria will be identified by macrophages. Macrophages are present in the lymph node. They will phagocytose the bacteria. Then they will be disintegrated. Their antigens will be released and they will enter in primary lymphatic nodule. Because of that, the lymphocytes will activate and they will proliferate and mature And finally, they give rise to two type of cells. These cells are plasma cells and memory B cell. What is the function of these plasma cells? They enter in medullary cord and release antibodies in medullary sinuses. The memory B cells, they have the memory of antigen. So they can identify antigen anywhere in the body. So they will go to any part of the body through blood or through lymph. So they will be circulated in the body. So they are coming out through the efferent lymphatics so that they can travel in the body, and wherever they will come across with that specific antigen, again they will get activated. So the basic function of the lymph node is filtration of the microbes, foreign material from the lymph, phagocytosis of this material, initiation of response, that is immune response, and formation of plasma cell and memory B cells. Lymphadenitis. In response to antigen, with proliferation of lymphocytes, the lymph node often gets enlarged. This is a common complication of microbial infection, and these lymph nodes can also be called as swollen glands. So we are done with the microscopic structure of the lymph node. Now we'll see the microscopic structure of thymus. It is a primary lymphoid organ, lobulated, encapsulated, present behind the sternum. Lymphoid stem cells from bone marrow are carried to thymus by blood circulation to form T lymphocytes. After the formation of T lymphocytes, they are being educated in the thymus. So basically, if we see the structure of thymus, now this is a diagram of a Chinese stained specimen of thymus, which we are trying to see under compound microscope under low power. You can see the capsule gives some fibrous septi. So the fibrous septa will pass inside the thymus to divide it into lobules. Each lobe is divided into outer cortex and inner medulla. So suppose this is one lobe, this is another lobe, like that. There are many lobes, and the lobes are again divided into outer cortex and inner medulla. The septa are incomplete, so the medulla of all the lobules is in continuity. So you can see the medulla is continuous. Lymphocytes are present in both cortex and medulla. So, which lymphocytes will be there? These will be the T lymphocytes. And they are supported by a network of epithelio-reticular cells. We can't make out the epithelio-reticular cells, but nuclei can be seen. Now we'll see the specifications of the cortex here. Each lobe has got its own cortex. In the cortex, the T lymphocytes are tightly packed. They are darkly stained and they will be small. So their nuclei are darkly stained. So these are all T lymphocytes present in the cortex. What are the features of medulla? Medulla of all the lobes is in continuity. So this is the medulla in continuity. The lymphocytes are loosely packed in medulla. As you can make out, the lymphocytes are wide apart from each other. They are lightly stained as it contains large lymphocytes with pale staining nuclei and more cytoplasm. And also in medulla, we can see epithelio-reticular cells. What else can we see in the medulla? The identifying feature of the thymus called as Hassel's capsules. So here you can see with green arrow, these bodies are called as Hassel's capsules. They are derived from type 6 epithelio-reticular cells. These are small rounded structure. They are made up of closely packed, concentrically arranged type 6 epithelio-reticular cells with flattened nuclei. So if you see the capsule, inside some amorphous substance is present surrounded by onion pill-like whorl-like arrangement of epithelio-reticular cells. If you see these Hassel's capsule under transmission electron microscope, you can see keratohyaline granules, intermediate filament bundles, and lipid droplets in these cells. The cells are connected by desmosome. The cells are connected here by desmosomes. Center of a thymic corpuscle may be keratinized, so sometimes keratin may be seen in the center. Few scientists say that these Husserl's corpuscles secrete interleukin that function in thymic differentiation and education of T lymphocytes. So we have seen the basic structure of the thymus under low power, how it appears. So it has got a cortex, it has got a medulla, it has got Husserl's corpuscle, it is covered by capsule, it is lobulated organ, and the main important thing here is blood is traveling through thymus and not the lymph. So there will be a barrier present between the lymphocytes and the blood entering inside, called as blood thymus barrier. What is the function of this barrier? Now, thymus is a primary lymphatic organ. That means the stem cells are differentiated into T lymphocytes without the stem cell being getting activated by antigen. So here the antigen should not come in contact with the stem cells and the T lymphocytes. So to protect the developing lymphocytes in the cortex from the exposure to antigen, we have a barrier called as blood thymic barrier. Let us try to understand this barrier. Now suppose this is the parenchyma of the thymus where you can see where stem cells or T lymphocytes are present and a blood vessel has entered inside the thymic parenchyma. So this is a capillary. So in between there is a blood inside. I forgot to draw the blood cells here. So there will be blood present inside and outside there are T lymphocytes. So here some antigens might be present into the blood capillary. So we want to prevent these antigens to enter into the thymic parenchyma. So what can we do? We have a barrier here. So what is this barrier made up of? First, endothelium of the capillary. Second, pericyte. Third, basal lamina of the capillary. Fourth, capillaries are surrounded by macrophages. And fifth, the supporting system of the thymus, that is epithelio-reticular cells. These cells are connected to each other by occlusion junction. So these all layers they prevent the contact between the antigens in the blood capillary and the lymphocytes present outside. This is called as blood thymus barrier. So we are done with the microscopic structure of thymus. Let us begin with spleen. Spleen is the largest lymphatic organ. It will filtrate blood. It removes and destroys senescent, damaged, abnormal RBCs and platelets. After that, it will retrieve the iron from the hemoglobin. It can help in formation of RBCs in early fetal life. In some species, it will store blood and basically RBCs. It reacts immunologically to the blood-borne antigens. That's why it is also a lymphoid organ. Actually, it is the largest lymphatic organ. To understand the microscopic structure of spleen, we again need to understand here the circulation of blood through the spleen. The way we saw circulation of lymph through the lymph node, here we need to understand the circulation of blood through the spleen. So let us try to understand this picture. As you know, spleen is supplied by splenic arteries which are entering through its hilum. Now the arteries will divide into arterioles. So those arterioles are entering into the spleen through the septi. So there is a septa here called as trabecula. So this splenic arteriole is entering inside and here it is entering in the splenic parenchyma called as central artery. Now why the name central artery? Because it is passing through the center of a sheath formed by T lymphocytes. This T lymphocyte sheath is called as periarterial lymphoid tissue or sheath. This periarterial lymphoid sheath, along with the central artery, now it enters into the basic architecture of spleen called as white pulp. Now, what is this? This is a lymphatic nodule present within the spleen. Here, this periarterial lymphatic sheath and the central artery are traveling little bit at the periphery of this nodule. So here, this is called as eccentric arteriole. This arteriole divides after coming out from the white pulp. It divides into peninsular arteries or straight arteriole. Now, from here onwards, the arterioles give out capillaries. So the capillaries are beginning here. The capillaries are surrounded by macrophages. They are called as sheath capillaries. Now there are two types of or two theories of splenic circulation. One is open circulation, the other one is closed circulation. In the open circulation, the capillaries are pouring the blood into the parenchyma of spleen called as splenic cords. So all the cells present in the blood are poured into the splenic cord. The second theory is closed circulation where these capillaries they open into closed venous channel called as splenic sinusoids. Now after this the venous system starts. So these will be the venules. The venules will come together to form trabecular veins, and finally the trabecular veins will carry blood from spleen to the splenic vein. So there will be formation of splenic vein. This is how the blood is circulating through the spleen. Now there is one more theory, a compromise theory may be present in human where both the circulation can be seen. When the spleen contracts, there will be open circulation. When the spleen relaxes, there will be closed circulation. So we just try to understand how the blood is traveling through the spleen and various names given to various blood vessels when they are entering into specific region of the spleen. Now when we actually see these things under H&E stain under a light microscope, how will it appear? We will try to see that. So basic structure of spleen is splenic parenchyma. So what you can see here, this is a low power view of H&E stain section of the spleen. A capsule is present. The capsule gives septi or trabeculae. The trabecular arteries are seen here, and inside there are lots of cells. So spleen is completely filled with cells, and there will be big dilated spaces. So these cells are lymphocytes. Basically, they are lymphocytes, and RBCs obviously will be present. In the fresh specimen of spleen, we can see two type of pulp, a white pulp and a red pulp. Now we will try to understand the white pulp. White pulp is a circular or elongated whitish gray colored area surrounded by red pulp. The white pulp is made up of cluster of lymphocytes surrounding a central artery. In hematoxylin and eosin stained sections, white pulp appears basophilic because of the dense heterochromatin in the nuclei of lymphocytes. So where is white pulp here in this section? This. You can see closely packed heterochromatin present in the lymphocytes, and lymphocytes are basically forming nodules here. But the specific structure of this nodule, they are having arteriole in them. Again, you can see arteriole only when the section is passing through the arteriole. Otherwise, it will just look like the lymphatic nodule present in the lymph node. So in this picture, at least you can make out one, two, three lymphatic nodules. So this is the white pulp of the spleen. The branches of splenic artery pierce the capsule. They enter in the splenic trabecula, dividing, forming trabecular artery. These arteries enter in the white pulp, divide, and they give rise to many central arteries. The T lymphocytes will surround the central artery to form periarterial lymphatic sheath. Periarterial lymphatic sheath is a thymus dependent zone. In a cross section, it appears circular. Then, this periarterial lymphatic sheath may be located in the lymphatic nodule at periphery close to germinal center. And then only you can see The central artery or periarterial lymphatic sheath eccentrically placed in the lymphatic nodule of the spleen. This is the identifying feature of the spleen. Now, the lymphatic nodule is predominantly formed by B lymphocytes here, and these are called as splenic nodules or Malpighian corpuscles. So, these are the white pulp called as Malpighian corpuscles. Few words about the red pulp. Red pulp contains red pulp contains large number of RBCs which will be filtered and degraded in the spleen. It also contains splenic sinusoids and splenic cords of Bellroth. So in this H&E stain section you can see the white pulp. In between white pulp, whatever you are able to see is the red pulp. In the red pulp, all the blood cells will be present. More number will be of RBCs and the splenic sinusoids will be present. But here you can't draw a sinusoid which has got a endothelium. So there will be sections of sinusoids. So in between I have tried to show the sections. These are the sinusoids present in between. Few words about the splenic cords or Billroth. They contain RBCs, lymphocytes, dendritic cells, plasma cells, granulocytes, macrophages. Splenic macrophages will phagocytose the damaged RBCs. Iron from these destroyed RBCs is used in formation of new RBCs. Splenic sinusoids are venous sinuses. They are lined by rod-shaped endothelial cells. Intercellular spaces between endothelial cells allow blood cells to pass readily into and out of the sinuses. Blood is present in both sinuses and cords of red pulp. So that was the basic architecture of the spleen. So we saw spleen has got a capsule, trabeculae, red pulp, white pulp. In the red pulp, we have splenic sinusoids and splenic cords of Billroth. In the white pulp, we have periarterial lymphatic sheath and eccentrically placed arteriole. Microscopic structure of palatine tonsil. Tonsil is a lymphatic organ present at the entry of oropharynx. It will have a medial surface and a lateral surface. The medial surface and the crypts are lined by stratified squamous non-keratinized epithelium. Underneath the epithelium, numerous lymphoid follicles are present. Lateral surface of the palatine tonsil is separated from underlying tissue by hemicapsule. Sometimes lateral to this hemicapsule, mucous salivary gland section can be seen. So when you see the slide of the palatine tonsil under low power in compound microscope, this is how it will appear. How can you identify it? On one side, capsule will be present. On other side, epithelium will be present. And in between, you will see lots of lymphatic nodule. So when you see some organ like this, definitely it will be a palatine tonsil. Now, what are the specifications? So laterally, the capsule is present. Medially, the oral surface is covered by oral mucosa. So the lining epithelium here will be stratified squamous non-keratinized epithelium. This mucosa invaginates and gives rise to many crypts. There will be around 10 to 20 crypts present in the tonsil, and one of them is very big called as intratonsillar crypt. Deep to that, this is the architecture of the tonsil where lots of lymphatic nodules will be present formed by T lymphocytes and B lymphocytes. So this is the basic architecture of the palatine tonsil. Little bit about the diffuse lymphatic tissue. The diffuse lymphatic tissue is seen in the lamina propria of mucosa of various structures in our body, where lymphocytes are diffusely arranged in the mucosa. The cells come in contact with antigens present on various target cells as bacteria and initiate an immune response. After contact with antigen, these lymphocytes enter in the regional lymph nodes, proliferate, and differentiate. Progeny of these cells, that means after formation of different cells from B lymphocytes and T lymphocytes, they return to lamina propria as effector cells and T lymphocytes. I hope you understood topic. Thank you for watching. Please do like, share and subscribe. Thank you.